Hello and welcome to our Lakeside chat. We are David Wofford and Katie Noonan, co-chairs of the Rotary Nature Center Friends. And we're so happy that you can join us tonight.
As we begin our program, it is important for us to acknowledge that we are on stolen land, that Lake Merritt is part of Ohlone territory. We hear now from Corinna Gould, spokesperson for the Confederated Villages of LaShawn and co-founder of Segura Te Land Trust.
Good afternoon, relatives. My name is Corinna Gould. I am the spokesperson for the Confederated Villages of LaShawn. We are here today at what most folks think of as Lake Merritt. We are in the territory of Huichin. Huichin is actually a territory that encompasses 6 Bay Area cities: Oakland, Berkeley, Alameda, Emeryville, Albany, and Piedmont. And so this is a place that my ancestors have been since the beginning of time. And this was a place of abundance. I'm so happy that people from all walks of life that now come into our territory can enjoy this beautiful place that my ancestors have enjoyed since the beginning of time. My relationship to the land that I have been born to, raising my children and grandchildren here, has been to tell the story, the truth of what happened on this land before other people came here. I'm hoping that as we begin to learn these lessons of fires in California, the pandemic that's happening, that human beings come back to living in reciprocity with the earth.
And I'd like to start by thanking all of the community members and organizations that have helped Rotary Nature Center Friends in the recent months to realize our mission, trying to bring people and nature together at Lake Merritt. In particular, we are thankful to the Alameda Countywide Clean Water Program that has made a donation to help us put on our California Naturalist course, and to all of the students, to Save the Bay, to iNaturalist, the wonderful San Francisco Estuary Partnership that put on the State of the Estuary program, Lake Merritt Breakfast Club, the City of Oakland, and Oakland Tech, Adopt a Spot, Environmental Services. There's so much out there supporting our community and our natural surroundings. Rotary Nature Center Friends has a focus of activity on community science and environmental justice, serving intergenerational local communities, all people who are come to Lake Merritt to enjoy it for various reasons, and we also participate in community science. So I'd like to show just a couple photos. Here is our California Naturalist certification class. We were able to take a geology field trip with Andrew Alden. It was a very special event, and it was beautiful. We attended the State of the Estuary Conference and presented a poster about the California Naturalist program and its goals in helping people who love nature to communicate better amongst themselves and with the public. And it was quite a party there. We had a wonderful time. And so now, at this point, I would like to introduce our speaker for tonight. And I know we're all waiting to hear from Damon Tighe, naturalist, scientist, educator, fungus fanatic, and it's going to be a wonderful night. I think many of us know Damon, but Damon was born in Northern California and went to college here. He taught high school for a bit and became a professional photographer, and also worked on the Human Genome Project. So he has a wonderful list of qualifications and accomplishments, and we're so lucky that we can have him here tonight to talk to us about UV fluorescent and bioluminescent organisms. What is that? We are so excited to hear about this topic. Damon, are you ready to?
Ready to go. Yep. All right. Sounds good. Good evening, everybody. Like Kay said, my name's Damon Tighe. I used to live right against Lake Merritt for, 2004 through basically most of the pandemic. And a lot of, the bioluminescence and UV fluorescence, or some of it you'll see in this talk, is actually directly from Lake Merritt. One way to think about both of these terms, bioluminescence, UV fluorescence, they're both kind of organisms producing light of some sort. One is by a chemical reaction, and one is by absorbing light and more or less letting go of the energy again at a different wavelength. But we'll dig into that in a little bit. Katie already gave you my background, so I can go ahead and skip this slide. Let's start with bioluminescence, because that's one of the ones that I think is the hallmark of Lake Merritt that a lot of people don't recognize, is that Lake Merritt has an amazing bioluminescence in the fall season. Tends to be around new moons in September and October. But bioluminescence, what is it? So most people's first recollection, if we ask them about bioluminescence, they think fireflies. And it's like, yep, that's a great thing to look at. So you've got this organism that is producing a reaction more or less in the hind portion of the beetle, and it's producing this bright light. So bioluminescence its derived kind of language is just an organism can produce light via an enzymatic reaction. That enzyme tends to be in the class of enzymes we call luciferases. And it's usually that luciferase plus a substrate and then oxygen. It's going to get you light and then some products at the end. And we have lots of organisms in California that do this. But one of the amazing things about bioluminescence, organisms that produce their own light, is a lot of times you can look at the wavelength of light, the color of the light, if you're thinking about like the visible spectrum, and that can tell you something about the taxonomy, which is kind of wild. So a lot of the stuff that ends up in kind of the blue and purple spectrum are things like shrimp, copepods that produce light, and things like that. Some of the dinoflagellates that we see in Lake Merritt also kind of operate in this kind of bluish zone. And then as you kind of move up the scale, fungi for the most part are producing in the kind of the green wavelengths of light. So all of our bioluminescent mushrooms in California, if you see them, they're popping out this really nice kind of deep green lights. And then of course, fireflies produce a green light, but it's even more skewed up towards kind of like green meets yellow, kind of, lime green zone. And then going even higher in the spectrum, when you get to things like click beetles and railroad worms, these other kind of like beetle groups, you'll see them produce in kind of this orangish to red level. We don't see too many of those in California, so that's mainly stuff you'll see in the tropics, but basically everybody else, you can easily see them in California if you know where to go look. So there is a project on iNaturalist, just to be aware of it, because if you're asking like, well, what around me glows, if you didn't make it to this talk or you can't remember everything, you can go to iNaturalist and type in bioluminescent organisms project. And you'll get to this project where I've been aggregating basically any observation that anybody has on iNaturalist of things that are producing their own light. And so it's really cool because you could just go to California, you could type into this project and then see exactly what glows here. Or someplace you're traveling, you could go kind of, pregame and look to see what might be there. One of my favorite examples of bioluminescence, like I said earlier, is Lake Merritt. I had always heard this kind of what I considered a myth at the time when I moved to Oakland about bioluminescence happening in Lake Merritt. And I had gone out quite a few times looking for it, had never seen it. And then I bumped into some rowers at some point who said that they'd seen it the previous fall. And I was like, well, if you ever see it again, you've got to let me know, because I want to be able to go out there and document it and see it and try and figure out what organism it is. And so late one night, I get this call from a rower friend, and they're like, it's happening. You got to get out there. And so I went out to the edge of the lake and kind of had just like a broomstick with me, swinging it around. And oh yeah, there's bioluminescence going on. And I was like, wow, this is really cool. I need to be out in the lake. And I didn't have access to a boat. So I called a couple friends, found a buddy with a canoe. We launched it and then basically went all across the lake trying to understand where it was to get an idea of maybe what it is. And at the time, I think we just hit it at peak. Basically, all the lake was glowing, but the biggest veins of it were like more or less from the Green Bridge heading right to the boathouse zone. You could hit these places where you put the oar in and it was just boom, super bright. In fact, it was bright enough that you can see in this photograph here. I mean, this is a picture at night with all that light pollution from downtown Oakland, and you can still pick up the blue there. Now, the crazy thing was I thought, cool, I'll be able to scoop some of this up, take it back, do some microscopy. We'll know who this is in no time, because I had no clue about the diversity of dinoflagellates that can produce bioluminescence. And so I ended up into the rabbit hole for quite a few weeks trying to figure this out, contacted every single lab I could find in California that worked bioluminescence. You know, I'm like, I've got okay microscopy of it. Can you help me out? And all we could do is get down to a genus level. But that also is also really exciting to know that you have a lot of marine organisms that are producing this bioluminescence, especially in brackish water systems. There seems to be quite a few of them. And by brackish water, I mean a place where you've got kind of salt water and fresh water meeting and mixing sometimes, which Lake Merritt does a lot of that, especially in the fall when we're getting those first rains coming down and we're getting, you know, the influence of the bay still there. You get a lot of kind of interesting brackish water interactions going on. So this is every year since that first event where I got to see it, I would go out religiously around basically September and October and stick the broomstick in the lake, go like this, see if I could get it to go. And then if I could see some fluorescence or some bioluminescence, I mean, I would jump in the water with a little inflatable kayak and then try and survey it. And so this is more or less the distribution of the bioluminescence that I saw through 2018, 19, and 20. And so the best time I've realized to see it tends to be September or October's new moon. And there's some reasons why we think new moons are best for this. So one theory is that these organisms, half the reason they do this bioluminescence is because they're blooming in numbers, but they also don't want to be eaten by other things like other small organisms. And so what they can do is they can basically light up small fish, like in Katie's lead up to this, right? In the questions, we had what fish kind of swims around with its mouth open. You might not want that swimming around with its mouth open, eating all of you and your friends. And so what could you do to get rid of that fish? You could light them up. And what's going to happen? It's going to be way easier for a predator fish to see that fish and then chase them off. And one of the things I have noticed when we do have the bioluminescent events in Lake Merritt, you will see a lot more nighttime fish jumping action in the lake. And so that can also be a key to be like, oh, maybe I should go check the water because I'm seeing a lot of stripers jump, which is kind of cool. Other bioluminescence that we have kind of like in the marine interface in California. Bodega Bay has some really good bioluminescent events. And these ones are a little bit different than Lake Merritt. I've tried going out in the water and I couldn't really get anything, but I'd heard reports from locals that you would get bioluminescence there. And one time I was going out there trying to go launch the little, inflatable raft, more or less. And I'm walking in the sand and I realized that my footsteps are bioluminescent, which is pretty wild. And so I took some of that sand home, washed it through and was able to find these dinoflagellates. And it appears this serratium is the thing that's making the bioluminescence in this case. And these are much, much larger than the genus of organisms that you're seeing in Lake Merritt. But the cool thing is you can basically go out to the beach there. And I believe this was October or November, and just streak your hand where kind of like the water is meeting the sand zone. And you just get these big, bright blue smears in the sand. You can also find, of course, larger organisms are producing bioluminescence in our region as well. So the dwarf brittle star, this is actually one I pulled off of a dock down at Jack London Square. And I forget how I came upon this, but I read an article at some point that these organisms produce light in order to scare off predators. They tend to be not, at the bottom of the dock, but like you're pulling up a rope that's down there quite a few feet. You'll find sponges. And these are kind of all infiltrated into the sponges there. And the theory is they produce light when like a fish or a crab tries to bite them, and they're like kind of scare you away with some light. And so I'd taken this one back and was like, well, how do I poke this thing enough to get it to produce light? And it didn't do anything when I was kind of poking it basically in a dish. But I had read some papers at one point that you could depolarize their neurons if you just dropped a bunch of potassium on them. And so I basically just took like a typical like potassium pill that we would take, or sorry, yeah, potassium pill that you would take, and just dissolved it in water and then put a drop on this guy, and it just boom, shot out light really, really brightly. So that's how I was able to get this photograph. Just had it basically on a slide in a dark box, drop potassium on there with my camera on it, and was able to kind of pick up that light reaction. And these are, I think these are non-native, if I remember right, to California. This is more or less from like the North Atlantic, but they're kind of all over the San Francisco Bay, especially if you get down into those sponges that are living a little bit deeper down in a little bit of the dark zones. Very, very ubiquitous over at Jack London on the, what I call like the free-for-all dock, the place you can pull up your boat or just lay there on your belly and look at stuff under the docks. Fireworms, we do have fireworms. Mainly, I see most of the observations from these coming from Southern California. And these, as their name kind of implies, they are a fiery worm, which means if you pick them up and you get stung, bit by them, it feels like there's fire burning in your hand. But they also produce their own kind of bioluminescence. I'm not really quite sure about the biology of why they're doing that, if this is to scare predators off of them. I mean, a lot of times if you're an annelid, you're kind of like, please, nobody eat me. I've got to have a strategy to deal with that. So I wouldn't be surprised if this is a bright flash of light to try and keep them away from being eaten. Other ones that we have closer to our area, sorry, too many things going on with Zoom here, is, and this is a photograph by one of our best kind of like amateur or community scientists, kind of marine folks, Robin, Nudibranch Mom on INAT, is these toadfish. And so they have bioluminescence on their belly, which is really, really cool. A lot of organisms that have bioluminescence on their belly tend to be things that have a predatory nature to them, that hunt at night. And so what they want to be able to do is go over prey without prey looking up and seeing a shadow. And so I think this is an organism that is probably doing that. The most famous one that does this, of course, is the bobtailed squid out from Hawaii. It's one of the ones that a lot of researchers do use the bacteria in them to do quorum sensing, and that squid is doing the same thing. It's a night hunter, and it doesn't want the prey to look up and see a shadow. It wants it to look up and see starry night sort of the situation. And so it has an organ that can basically get Vibrio bacteria in them, get to a concentration when they get to quorum density, and then they'll produce light. And so when the prey looks up, they see a starry night before getting eaten. These guys you can find in intertidal zones. And so it's one of the ones to kind of look out for if you're going out on those low tide events at Pillar Point and places south. I know everybody's always like, does California have fireflies? Yes, we have fireflies, but we don't have fireflies in the way that the East Coast thinks about fireflies, or South Africa, but South Asia thinks about them, or even South America. None of our adult fireflies, which are actually beetles, none of them produce light as far as I know, but the juveniles produce light. And so this is one of the larval stages of one of our fireflies, the California pink glowworm, and it has extremely, extremely bright bioluminescence. My first encounter with this was on the PCT outside of Lassen. Me and my buddy were doing a night hike section of the PCT just to kind of make up miles. And we had stopped by a firm oil, which is already kind of a spooky place to be. There's steam coming out of the ground. And we kept seeing this light in the distance. My friend was convinced that it was somebody like stalking us, and he started yelling at it. And I was like, I think no, I think it might be something like biological. We went over there and we found just one of these producing this extremely bright light. People do spot these in the Bay Area. I have not been lucky enough to see them, but I definitely see observations of them in iNaturalist, predominantly from the South Bay, the forest kind of in the South Bay leading over into the Santa Cruz Mountains. But these are definitely around. Why would you want to produce light as a larval stage? Well, a lot of these larvae like to eat other insects. So it's a way to kind of trick other insects to come, whoa, what's that light? And then you get to eat them, which is a classic kind of mechanism that you see a lot of predatory insects use when it comes to bioluminescence. They'll use it as a way to draw in small prey to then eat. So like the glowworms of New Zealand, it's the same thing. They're a fly larva that does that. Now into my specialty, into the fungi. We've got quite a few fungi that have bioluminescence in California. Unfortunately, none of them are like the tropics necessarily, where they're super bright and it is actually the mushroom producing the lights. A lot of times, the fungi in our area, it is their mycelium producing the light, which brings up all kinds of interesting questions of like, why? What benefit is this? And there's lots of interesting hypotheses. Some of it may be that it's actually trying to appeal to wood-boring insects that will then get covered in mycelium and then take that mycelium to other host substrates. It may be just a way to just get rid of energy, like in the same way that you might use heat, to dissipate extra energy you've got. Some people are suggesting that might just be getting rid of the energy in the form of like heat and photons is a great way to do it. But one of the ones that's really easy to find but hard to see glow is the bleeding fairy helmet. And so this is a classic mycena that we see on a lot of the oak logs in the SF Bay. And the reason it's given this name is if you scratch the cap with a blade, it will produce what looks like blood coming out of the top of the mushroom cap. But if you take some of that wood home and you basically just cut it at night, you'll see the wood glow. And there's probably a number of the mycenas in California that have bioluminescence like this, but they just have not been documented. So this one at the time I took this photograph with hypothesized that it might do this because it was in a group that we know to do this. And this, I think, was the first document of it. It was this kind of crappy photo, but it's science. You're like, I got the document, good to move on. But I really encourage people that are interested in fungi to try to do more work with some of the mycena in our area, looking for bioluminescence in the wood. And this would be like cut the wood, take like a long time exposure. I think this was like a 5-minute exposure. Dripping bonnets are another one that have nice bioluminescence. These ones you tend to find kind of like North Bay and going up to the Oregon border. These ones have a slime veil on them and just the stipe glows, which is really kind of weird and interesting because the slime veil we think is used to basically trap insects so they don't go up and eat the gills. And the glow tends to happen right where the slime is. And so it might be a way to get these little tiny flies and springtails that are eating your gills to go to the sticky mess, trap them so they don't eat your gills. But this is a pretty common mushroom, especially once we get our first kind of cold rain. These will pop up in good numbers all over the place. You can also see this one has bioluminescence in the mycelium as well. So this is another one of those rhodomyces. And this is the substrate that it's on, which is just some like broken up woody material, some leaves mixed in, and that had a nice resonant glow to it. Purple edged bonnet, closely related to bleeding fairy helmet, also does the same thing as a bleeding fairy helmet. It'll have a nice bioluminescence if you cut the wood or cut its substrate. This is the most common purple mushroom you will find on pine cones in our area, so it's really easy to identify. And then the other way to identify it is the gill edges will have this nice kind of like pink edge to it, what we call a marginite gill color. But if you're to cut the cone or cut the wood, you'll see bioluminescence in there. Bulbous bonnet also has that glow. These are some photographs that Alan Rockefeller took over at Point Reyes. But you're kind of getting the theme here. A lot of these mycenas have bioluminescence in them. And some of these you can see at night if you're in like a wet enough forest in California. Sometimes you'll see actually just the substrate as you're walking a trail glowing. Honey mushrooms are a notorious mushroom for killing other things. Like I think Guinness Book of World Records has them listed as the largest organism on the planet. There's one in Eastern Oregon, I think it's like 15 kilometers in diameter, at least the mycelial map from it. But interestingly enough, when they're busy killing their host trees, that mycelium often glows. And so I've had lots of reports from the Sierra foothills of friends where I grew up taking out trees, and they were taking them out like around sunset to avoid like when heat is. And they would be like, the stump is glowing as I'm trying to remove it. And I was like, oh, have you seen honey mushrooms there in the winter? And they're like, yeah. And it's like, oh, that's what it is. So the mycelium on this one glows really, really bright. And we've got a number of honey mushrooms in California. I think this is the only one that we have a nice documented observation of having bioluminescence. So this is Armillaria sinapina, which is one of the more common ones that you'll see in the Bay Area, especially if you get to kind of the coastal side of the Bay. It's very, very common early in our mushroom season. So right now it's actually up. Our biggest and brightest bioluminescent mushroom, though, is the Western jack-o'-lantern. And as the name suggests, it's kind of as bright as a jack-o'-lantern sometimes. It also happens to usually occur right around Halloween. So I think that's part of the name, part of the reason it gets this name. These ones are a very large fruiting body. And by large, I mean, they can be the size of my head. And when they are glowing, which isn't always, I've looked at quite a few of them, and like the range of when they're glowing is all over the place. But when you find one that's glowing, I mean, it is incredible. You can see it from, yards away. So this was a cluster that I saw out in Martinez after a hike I had led for people. And it was just extremely bright. Like I could see like the shadow of a tree, after sunset, and I could just see this green glow over there. So I went and kind of set up my camera and took this photograph. And the one on the top right is just one from Mount Diablo, where I couldn't see a glow in the field, but brought it home and photographed it, and then could easily see the glow. Southern California gets the giantest ones I've ever seen. These really like to grow on dead oak stumps or dying oaks. And they are up at whenever there is moisture, more or less. So they're just kind of starting right now. And then they'll kind of go through usually January. Once we start to get those really, really cold nights, they kind of start to drop off. But a really easy to spot mushroom because of their size. And then, your best luck is actually just kind of pull one or two of them, take them home, and then take them to a dark closet, wait for your eyes to adjust, and then see if they glow, or train your camera on it. They also happen to be a mushroom that produces a really, really nice dye. And so they're kind of a coveted mushroom by people that are into natural dyes. Just using vinegar, you can just crush them up with some heat, and they produce this really, really nice kind of lavender color dye. This is actually some dye that some high school students that I was working with in Morro Bay made once while I was doing a talk. We have one mushroom in California that, man, I wish it glowed more consistently. So the bitter oyster, also known as Panella stypticus, is this really, really well-known glowing mushroom that you see in Europe and on the East Coast. In fact, I just saw some this past week. I was in St. Louis, and I just took a little cluster home, went into the hotel room, barely needed to turn off the lights, and I could see them glow. In California, I've looked at about 20 specimens, taking them home, photographed them, never been able to see glow. And kind of the lore in the mycology community is that they just don't glow once you get kind of west of the Rockies. But Alan Rockefeller has this photograph from, I think this is Point Reyes, of some glowing. So there is hope. They might just not be as strong as the ones from the East Coast. Our guess with a lot of these mushrooms that are glowing at night in the fruiting body is that they are appealing to insects. So they're producing light in a wavelength that some of these insects perceive. Insects will fly over, investigating the light, and then they'll get basically covered in spores. And then they'll fly away with those spores and basically deposit them someplace else. From what I can tell, decent theory. We could definitely use some more evidence on it for specific species. We do have an interesting group of bioluminescent millipedes in California. And I'm still kicking myself because I found one in SLO County, but didn't know it was bioluminescent and was just obsessed with how UV fluorescent it was, and didn't get a photograph of it doing bioluminescence. But for the most part, this bioluminescent clade of millipedes, flatback millipedes, tends to be in the Southern Sierra. And some of the photographs I've seen of them are just mind-blowing. And they are an interesting group where I have no clue why they're doing the bioluminescence. I also have no clue why they do the UV fluorescence, but both ways that they produce light is very, very bright. And actually, unfortunately, I don't have the photograph of it here, but if you do go on to iNaturalist and you look into Mytixia, this group in the Southern Sierras, you'll see one of the researchers who studies this group from Tennessee has a picture of them in his hands. And I mean, it's like lighting up his face. It is incredible. But one more thing to look for than Sierras, which are just an amazing place, right? So much going on there. So let's switch gears. Since we've got this kind of organism that does both things, it can produce light through an enzymatic reaction and it can do UV fluorescence. Let's talk about what UV fluorescence is. UV fluorescence is when you are able to absorb light, usually of like a high intensity light, UV in this case. And what happens is you take that light in and then you get rid of that energy by usually putting off a little bit of heat probably, and then letting go of a photon of light in a different wavelength, something in the visible spectrum. So it takes light that we can't see, and then it transforms it into light that we can see. And if you're an organic chemist or more in the chemistry world, this is actually the way you're thinking about it. It's coming in. This is if you're not a PChem person. It's coming in. It's hitting an electron in the electron shields, and it's bumping them up an orbital of energy. And then what's happening is the electrons do not like to be up there, and so it's not a stable place to be. And so they'll drop back down to their stable orbit. And when they drop back down the stable orbit, that's how they're letting go of that photon of light. They're getting rid of energy in that way. And that photon happens to be in the visible spectrum. And so there's lots of compounds that can do this sort of modulation. And to be honest, like this is still an emerging field. So we know a handful of these. But the diversity of things that we see fluorescent in California leave me kind of like, I wish we knew more. So some of the common compounds that are really well known to do UV fluorescence that you might have interaction with are things like curcumin. So you'd find that in turmeric. If you've ever cooked with turmeric before, I would encourage you to then take a UV light and look at any place you've cooked, and what you'll realize is you have this bright, glowing orange yellow dots everywhere from curcumin getting into oil and the oil ending up on stuff. Beta carboline is another thing that you'll find like in exoskeletons of a lot of arthropods. So you'll see it in scorpions. This is one of the things that causes them to have that really kind of bright green fluorescence. Basically, anything with a kind of a benzene ring in the structure seems to be able to modulate UV light coming in and then turning it into a different spectrum of visible light. There is an iNaturalist project for collecting bioluminescence. And this one has been, I mean, it's been huge. A lot of people contribute to this from all over the world. And it's really, really fun because you can kind of like test hypotheses out using this. I've been using it to like encourage people to look at specific groups so we can get more information about them. It's really kind of a fun Easter egg hunt at this point, especially because this hasn't been something that's been studied incredibly well within kind of academic science, because just access to UV light, especially in the field, has not been like ubiquitous. And that's mainly because previous to kind of LED technologies, you kind of had to have like practically a generator with you or a lot of battery power to then power, a way to get that UV spectrum out in the field. So it was really only kind of like geologists and then their sidekicks, the scorpion guys that had these big lights and were going out and looking at stuff. But now that you can buy, a pretty inexpensive light, like off the internet for like $30 is the one I run with, like you can see all these things UV fluorescing and get an idea of like the diversity of stuff that does this. So we'll jump into some of them. Barred owls, a lot of birds will have UV fluorescence. And interestingly, it seems that owls specifically will accumulate something that causes stronger UV fluorescence over the course of their life. And so you can actually age birds or owls specifically with a UV torch relatively quickly. And then also females appear to produce more UV fluorescence than males do. So you can kind of like age them and sex them relatively quickly using a UV light in the field. One thing that's interesting is when I've hit birds in the field at night with the UV light, all of them react to it. They'll move really quickly, where a lot of organisms don't seem bothered by the presence of UV light, which makes me think that there's some sort of biological function to this for them. Like they're able to pick up on the UV fluorescence or something there. But these are some photographs that somebody had from a lab that was studying birds. I actually don't have many good photographs. I'm just not a birder. I'm not capturing birds. But if you're somebody that works with birds, I would really encourage you to get a UV torch and just start doing just some, cursory looks at the wings for UV fluorescence. One thing I do get a lot of in my yard, though, that I can easily get my hands on is possums. I've got like a resident possum that hangs out in the yard and is very chill with me coming up to it and putting lights on it. And possums have amazing UV fluorescence. This photograph doesn't do it justice, but that entire body basically is hot pink. And then right around the face and the tail, you'll get a blue fluorescence. And there's some studies that have shown within some nocturnal mammals, possum's not of course a mammal, but that you can actually see individuals based upon their fluorescence patterns. And I've seen that a little bit with the possums in my area. I've got like one old mama that comes through all the time, and I've got a couple of youngsters that'll come through the yard, and they all have different kind of blue fluorescence patterns on the face. What's causing this? Still don't know. My guess is it's probably maybe something in the fats that are being produced in their skin. But open question, but a really, really amazingly bright organism under a UV torch. Fungi, of course, have a lot of UV fluorescence. One of my favorite ones is bird nest fungi, and that's mainly because they don't fluoresce easily if you're just kind of looking at their external structure. But if you cut them, the little petioles inside, with our little eggs per se, in the bird's nest fungi will have strong, strong UV fluorescence. And you'll see this with different fungi, is that different tissues have an enormous amount of UV fluorescence and then others don't. And my gut feeling here is that a lot of the things that are producing UV fluorescence, they tend to be things where the spores are being produced. And so they are modulating light to get some sort of vector to come through and then move the spores, most likely an insect. And one thing to remember with UV fluorescence, I know a lot of people will see it and be like, UV fluorescence, when I see what I'm seeing right now, and it's like glowing back at me, that must be what insects see. And it's like, not quite. What we're seeing is that there's something there that's absorbing the UV light and then getting it reflected back at us at a different wavelength, right? So it might tell us the presence of molecules that are absorbing UV light, but it's probably not what the insects are seeing. And I've got a slide on that in a bit that can kind of drill into that a little bit more. Gymnopolis mushrooms are something that glow extremely bright. All the Gymnopolis mushrooms you can find in the San Francisco Bay have very, very strong UV fluorescence. This specific Gymnopolis, though, is kind of interesting because it is one that is, I feel like, impossible to see without a UV torch. Like you literally have to be digging apart redwood stumps in order to find it on its own usually. But if you walk through a forest with a UV torch at night, you can pick them up, I mean, really, really quick because it is such a striking green color. It's like seeing a scorpion at night. Powder caps, Cystoderma is a group that has some really, really nice kind of like fairy-like, UV fluorescence on the gills. It's interesting because it doesn't necessarily give you like a bright color, but it just gives you this bright kind of like aura in kind of a little bit of a pale green spectrum, but just a very, very stunning mushroom under UV light. Cystoderma is something that you'll mainly find in really, really wet, heavy duff. And so I would say like the edge of some of our redwood forests where they start to interact with oaks is some of the best places to look for these. Schizophilum commune, which is a notorious mushroom in the mycology world because it has 23,000 mating types, which just means it has a lot of different partners it can choose in life in order to do fungal sex with. Has unusual fluorescence. So we don't really see blue fluorescence a lot with mushrooms, or at least a really deep blue fluorescence, but this has a very, very strikingly blue fluorescence on its gill edges. It doesn't have any fluorescence any other place on the mushroom. When I've tried dissecting it to understand like what is producing the blue, when you get into the gill, in some of the cells that are on the gill, I can find what looks like oil droplets inside of them. And it appears to be that these oil droplets is what contains whatever is doing the blue fluorescence. I really would like to work with somebody ultimately to kind of extract that material, mass spec it or something in order to figure out what it is that's producing that blue fluorescence. Entoloma subcarneum is also one that has just extremely beautiful blue fluorescence, but like once again, very unusual to see strikingly deep blue fluorescence on mushrooms. This is something we don't see necessarily in the Bay Area, but once you get to Mendocino and north, you can run across this very, very beautiful mushroom occasionally. Sulfur tufts are something you can see all across California. I find them at Lake Merritt, and this is easily one of the strongest UV fluorescent fungi you'll ever find. Most of the Hyphiloma genus is just like a bright star at night with a UV torch. You can walk through a forest and just be shining like this, and at about 50 feet, you'll be able to see them really, really obviously. So these ones are unusual for some fungi in that the caps will fluoresce, the gills will fluoresce, and the stipes will fluoresce. And it all produces kind of like somewhere between like a bright green with a little bit of a blue shift in it. So this here is a photograph with UV light on one side and then white light on the other, so that you can understand what they look like in both spectrums. These tend to be around areas where you have a lot of decomposition of wood. So if you've got rotten stumps or you've got logs that are down, these tend to be in the soils right next to that. And these are one of the earlier fungi that come up. So since we just had that rain, like this next week, these will start to pop up, and they'll be up for probably 3 to 4 months. They're around for quite a while. So here's another shot of them. You can see how kind of like bright they are. We do have Cortinarius mushrooms that are also unusual, and that the whole thing is just extremely bright. And you'll be walking through the forest and not be able to see them during daylight because they blend in so well. But you walk out with a UV torch and it's like, whoa, wow, that is striking. I see these pretty frequently over in Marin County. They're very ubiquitous once you get up into Mendocino. They tend to be in the bay oak meets a little bit of pine. So in that really, really kind of mixed forest is where they like to hang out. Very, very striking mushroom. Insect egg slime mold. So we do have slime molds that will have fluorescence, but the weird thing about fluorescence in slime molds is it very, 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 very rarely is actually the fruiting bodies of slime molds that will fluoresce. But the plasmodial slime kind of stage of them is what will have the strong fluorescence. And so insect egg slime mold is one that you can pick up pretty quickly if you're walking through a forest at night, and you'll see this kind of like weird kind of glow just in all of the duff. It's usually this. And then if you come back in a couple of days, then you'll see the insect eggs, the kind of fruiting body of these all over the place. Lichens are, of course, a classic thing with UV fluorescence. In California, I always kind of expected that the desert would be the place to be where there'd be lots of UV fluorescence. And yeah, you do see ones on rocks there that have lots of UV fluorescence. But in our forests, we also have quite a few of them that will be in the deep forest that will have strong UV fluorescence. And the reason I thought that you'd mainly see them in the desert zone is because they're getting bombarded by UV light all the time, right? And so as an organism that's getting bombarded by UV light, you want to be able to protect your DNA and your proteins from that. And so you would accumulate a lot of these molecules that could absorb UV light and then do something with it, right? Get rid of the energy in a different wavelength. But I think what's going on in our deep forest, and this is by going back at different times of the year, is really what we're seeing is some of the acids that are produced in these lichens coming out that have the UV fluorescence. And so if you go to one of our forests within the first couple of rains, I mean, boy, if you go on the first rain, I mean, you're basically stepping into Avatar. every lichen-filled tree you see will just be super bright because everybody has had all these metabolites building up and they are getting pumped out, with that first kind of pop of moisture. And then as the season goes on, they kind of decline a little bit, and there's certain species that will be bright, but you still have ones that are doing UV fluorescence throughout the entire year in the forest. But boy, if you can get out on that first rain, do it. Go to a nice kind of like old growthy, California live oak forest, and it's pretty incredible. Tube lichens are one of my favorite ones for bioluminescence, and that's mainly because they have multiple colors on them. You'll get this nice kind of orange glow, and then where they're kind of spore producing kind of pieces, you'll get these, or the eyes, you'll get these bright red glows on them. My gut feeling is that red that you're seeing there is probably from the chlorophyll, since chlorophyll A produces red light when you shine a UV light on it. And you'll start seeing this in plants if you go out the UV torch. Every single plant you hit will have red fluorescence on it. Some of our mollusks will also have really kind of surprising UV fluorescence. So some of our nudibranchs, like the Modus Catalina here, you hit that with a UV torch and it goes from being yellow to really, really cool blue. Intertidal zones are some of the coolest places you can go with a UV torch because a lot of things there are dealing with UV coming from an evolutionary history where they may not have had to deal with it much. They've produced a lot of these compounds to kind of block, I get exposed to UV at some point because of the tide change. Other mollusks, of course, ones that are rolling around our forests also produce UV fluorescence. So a lot of the snails you find in California, you hit them with a torch and they will be UV fluorescent, usually in the blue spectrum as well. Chitins, also will have UV fluorescence in that intertidal zone, and these can be really, really, really striking. And we have got some gorgeous chitins just under white light in California, and then you hit them with a UV torch, and it's just through the roof cool. And of course, the classic one that everybody thinks about with UV fluorescence, that's why I kind of buried it in the deck a bit, are things like scorpions. So we've got a number of scorpions in California. All of them do UV fluorescence. And they've got like an interesting kind of history on this because this is something that's actually been studied for a while because this is some of the first kind of like UV fluorescent organisms that academics really got, intrigued by. So here's the one you can find easily in the San Francisco Bay, which is the California forest scorpion, or the best metal band name, if you're looking for one, Euroctonus mordax. What a researcher in the University of Arizona realized was that through a lot of experimentation, that the fluorescence we see from scorpions is being used by the scorpion to understand when they are exposed. And so what will take place is that when they've got a body part exposed, even just that little bit of moonlight they're getting at night gives them enough fluorescence that they can see it and then back up. And the wild thing is it's not their eyes. So this researcher basically painted their eyes shut or blacked out their eyes and then hit them with a UV torch and then realized they would still back up. They'd still go into a dark place. And he's like, oh, that's really kind of interesting. And what they realized is that they've got extraocular sensors on the tail that can pick up the fluorescence. And so if you were to go over with a green light in that same spectrum that you're getting the UV fluorescence in, the scorpion will also back up. And so those extraocular receptors apparently pick up that green spectrum and are like, hey, you're exposed, time to back up and get into a safe place. Also, one thing just to be aware of, if you get a UV torch and you start walking out of our forest, you'll realize, like everybody does, that like, oh my God, there are scorpions to be found basically every place on the planet, except for maybe Antarctica. These ones here are photographs from just our hills. So I think this was Joaquin Miller at some point. Golden crab spiders, and most of our crab spiders will have bright fluorescence. These are ones that hang out in flowers, and they are probably modulating the light in order to either draw attention to themselves and be like, hey, look at me, I look like part of the flower, or maybe they're modulating the light to kind of like blend into the background and be like, you don't see me, and now I can grab you. But a fair number of the crab spiders that I've encountered in California have bright fluorescence. There is a paper of some of these crab spiders that don't hang out in flowers, but will hang out on plants, that they apparently are just pretending to be a flower on their own. Like, hey, look at me, I look like a flower. You come by to like get some pollen and I'm going to eat you. Wind scorpions, which you can find in the San Francisco Bay, also have just really cool UV fluorescence. It tends to be on kind of the hairs. And if you get lucky enough to find one that is really big, they're basically see-through with the UV light. And so you can see all the fat globules inside of them, all these fat deposits inside of them. Really, really cool organisms under a UV light or in white light. A lot of our arachnids have UV fluorescence. A lot of our kind of ground-dwelling spiders like Calosoga, the false tarantula, will mainly have fluorescence right around the joints and then in the eye zone. I don't have a good theory for why this is, or why you would want to have that, but this may be one of these things where it's just an artifact and not, a biologically driven thing that there's evolutionary pressure on. Things that do have a lot of fluorescence, so in the arachnid group, tend to be things that make webs. A lot of our web spinners, like the cat-faced spider, which is out in big numbers right now, and our pumpkin spiders and things like that, any of these orb weavers tend to have really, really bright fluorescence and then all across the body, which to me makes it seem like they're probably trying to be like, hey, insects, come over here. There's something here for you to look at. And they get stuck in the web and then eaten. Some of the ornate checker beetles, which you'll find in a lot of like aster flowers and things like that, will also have really, really strong UV fluorescence. I haven't seen any papers on why they might be doing this, especially because I'm like, you're not trying to hunt anybody, but maybe this is their way of like blending into the flower, maybe to predatory birds that might be looking for them. I don't know. Sea anemones, lots of UV fluorescence there. All of our sea anemones in California, from what I've seen, glow stupendously. So here's the moon glow, which has this really, really kind of cool, like lavender pink color under a black light. The strawberry anemone has UV fluorescence, but really right only around the mouth part, which is super cool. I'm not really sure what's going on there with that. And of course, our giant green sea anemone is just, it's one of these things you can see at a huge distance with a UV torch. And they will just glow they're bright green normally, right? And under the UV light, it's like that in disco mode, just super, super bright. And a lot of the nidarians, things like sea anemones, right, have proteins in them that can actually modulate UV light and then turn it into a different spectrum. In fact, one of the ones in Aqua Victoria, which is the crystal jellyfish, which we've seen a couple times in the bay, this actually is where the green fluorescent protein that I use all the time in my day job in biotech originally came from. And this GFP protein was basically a major, major breakthrough in biological sciences because it allowed us to understand. You could basically stick it next to a gene and understand when it was being turned on because you could put a light on it and be like, oh, it's glowing now. I guess that gene is on. So it has at least 3 or 4 like Nobel Prizes where this has either been the foundational thing or a tool used in order to understand the biology of things. Flatback millipedes, like I said before, have lots of UV fluorescence. They're just starting to wake up right now in the Oakland Hills. So if you go out there at night they tend to like to be in kind of like semi-open zones next to forest. They're debris eaters. So they'll just be going through munching leaves and other kind of plants that are wilting right now. Occasionally you'll go out and you'll just see like 100 of them in one zone. They're kind of odd because they don't really respond to the UV light. They're not like a scorpion. They're not like a bird where like they get hit by the UV light and they walk away. They just go along, prancing along with their bright selves. Plants, like I said, a lot of plants will have UV fluorescence due to chlorophyll A. Chlorophyll A can absorb light and then kick it back, or the UV light, and then kick it back out in the red spectrum. And that's what you see here on the Mediterranean milk thistle. But the weird thing about the Mediterranean milk thistle, a non-native, is that the cotyledons are bright, bright blue. And I'm not sure what's causing that, but as soon as those cotyledons are gone, just the rest of plants is bright red. We see a little bit of blue on some of the other thistles, but I'm not sure what drives it. So some of you were asking what's a good like UV light to go out and start playing around with this. There's kind of two ones that you can find really easily online. There's PET urine lights, which will run you about 10, 15 bucks. And these tend to be in the 395 nanometer range, which are great. This is where I started originally. But the only problem with these is you get a lot of like purple and blue light that comes from that LED. So you'll see fluorescence, but it's kind of obscured a bit due to all this other colored light that you're seeing. And then recently in the last like 3 or 4 years, these kind of like higher end lights came out. Like I think the first one I bought was like 80 bucks, but now they're down to about 30 bucks. And they are producing light at 365 nanometers, so a little bit more energy. So a lot more things will fluoresce because you're hitting with more energy. It's still in the safe zone. Like it's not like you're going to shine it on yourself and damage your DNA or your proteins. You have to get down beneath like 300 nanometers, 280 is really where you start to worry about stuff. But they also have a filter in them that gets rid of the blue and purple spectrum from the LED. So all you see is the fluorescence. And that's like half the reason to get one of these. And the way you find these is you can type in C8 is kind of like the original torch that was doing this. So if you type in that into something like Amazon, you can find all the knockoffs, and those are like 30 bucks or so. If you end up with a PET urine light, or you've got one that's producing a lot of purple or blue spectrum, if you just put on sunglasses that are yellow, it will get rid of that light for you, and then you'll only be seeing the UV fluorescence. So when I just had my PET urine light, that's what I would use a lot, is just range glasses, and then the whole forest like lights up with that PET urine light. A lot of plants in California have UV fluorescence, especially in the flowers. So of course the leaves are going to be red because of chlorophyll. But what you'll start to realize is petals are modified leaves. So like our California corn lily, for example, is a white flower, but if you hit it with a UV light, you see red. And why are you seeing red? Because there's chlorophyll there, because petals are modified leaves. And then a lot of times what you'll see in a lot of the California plants is that pollen will fluoresce very brightly. And then occasionally the ovary will fluoresce really brightly as well. Sometimes the nectaries will also fluoresce really, really bright. And then why you'd have a nectary that fluoresce, right, is you're trying to attract insects in. So there's something going on with UV modulation there. But once again, just to remind us that UV fluorescence is not what insects see. So this is a great series that I got from a friend in Texas. Here's white light on the left. Here's UV fluorescence in the center, which everybody gets excited about because you feel like you're on drugs looking at it. But then really what insects are seeing is this on the right. This is UV spectrum. And so this is a camera that picks up only kind of reflected UV light. And then Michelle Wong is using a flash here with a lot of UV spectrum. So we get back with what the insects see. And this, kind of paints the story, which is like, hey, now I've got a target of a place I should go, right? Versus this, which is there's a lot going on. And it's really pretty, but it's not as kind of directed. A lot of our flowers have weird modulation of light in California. Our fritillarias have that. Our trilexia or our pretty face has that. Snow plants have it. Lupins, some of the lupins are incredible under UV light, but it tends to not be the flower so much as like the flower stalk and the hairs on them modulate UV light really, really intensely. California pipevine, not too surprisingly, the whole dang flower-like structure, right, is bright red under UV light. Showy milkweed, these are ones that you can find at Lake Merritt that somebody guerrilla planted. They are incredible under UV light. It's like a starry night. If you want to follow people that do a lot of this, these are kind of people that contribute to this slide deck that have a lot of like UV fluorescence and bioluminescence data in iNaturalist. So they're just kind of good people to kind of keep an eye on if you're looking to understand more about this. And then if you're interested, this link right there, we get you to the slide deck. I can also drop that into the chat. And I know we're running right up against the hour, so I'm going to go ahead and stop the share at this point. And then we'll get to the questions after Katie does a little wind down. Katie, you're on mute.
I was trying to keep my dog quiet. Okay. Yes, thank you so much, Damon. This is so fascinating. I know we have lots of questions. I'd like to take a few minutes to thank people and to give our credits. Okay, so I want to thank everybody for coming tonight, and hope to see you in December when we will have another Lakeside Chat. We want to give special thanks to Damon for a wonderful and just a brilliant presentation. To our new voices are rising high school externs and fellows, California Naturalist class, Save San Francisco Bay, iNaturalist, of course, many other organizations that we mentioned in a previous slide. We're so grateful to have the support and involvement of the community. Coming up on Friday, December 7th, Lakeside Sat 61 will be with Eli Kirsch, the CEO of the Lake Tech Company, who has been putting both water quality monitoring buoys into the lake since since September of 2022, right after the fish kill, and continuing with the City of Oakland Lake Merritt Water Quality Management Program. He's going to give a talk to us and bring us up to date on what they found out and what the options are for the future. And so we're looking forward to having that update from Eli. We will be doing Adopt a Spot mornings in the marsh, water testing and plankton viewing as we have time in the coming weeks. And the place to find out about that would be our Facebook, which we publicize what we do and possibilities to join in with us as they come up. And just noting that Martin Luther King Day of Service is also coming up, and we'll be participating in that. The Lakeside Chats are recorded, of course, and are rebroadcast on the City of Oakland KTOP TV. So the first two Sunday evenings from 6 to 7 p.m. are devoted to golden oldie programs, and the last two are devoted to the current program of the month. So good place to share our what we've done and to check in with past shows. We want to give another shout out for our new Lake Merritt Wildlife Calendar. This is all new with new species and new photos. It's going to be very exciting. We'll show you more in time for the holidays. It's going to be really lovely. If you like what we do, please consider supporting Rotary Nature Center Friends. We're an all-volunteer group, and everything that we receive, we use and put into program and to bringing things like Lakeside Chat and so forth. We want to thank our producer, Rob Lamone, for media and videos. I'm Katie Noonan, and David Wofford's with us tonight, co-chairs of Rotary Nature Center Friends. As stewards of the Lake Merritt Wildlife Refuge, we regularly host groups and members of the general public for bird watching, nature walks, plant identification, and more. So we encourage you to check us out at Lake Merritt. And again, really happy that you were here tonight, and really thank Damon for his wonderful presentation. And now we're ready for all of your wonderful questions. So let's see.
So I was trying to reply to a couple of them in the chat while doing that. So I might just read them off, those ones I already kind of just jumped in on.
Yeah, because other people will be more interested.
So Anthony had asked, does bioluminescence produce fall into the light spectrum that humans can see, or is bioluminescence that falls into the UV range that may be seen only by those insects, mammals, etc.? My gut feeling is there probably is bioluminescence being produced outside of our visual spectrum. And I would be surprised if it's produced into the UV spectrum, just because UV is a lot of energy to produce. But I wouldn't be surprised if going out the other direction on the spectrum, if there's light being produced there that we're not seeing. Also, intensity is a really big thing here, right? So the ones that we're seeing are also just being produced at an intensity level that we can pick it up with our eyes, because everything produces light, but can you detect it? So even like a human body right now, I am actually pumping out photons, but all of us don't really have the ocular system to pick up those like minor number of photons that are being pumped out by my metabolism in my cells. But scientists have built optical systems for being able to pick this type of like really, really low amount of production of photons up. So there's an intensity question, I would say, in all of this, and a what portion of the spectrum. So my gut feeling is understanding other organisms' visual systems is probably the linchpin of really understanding this further. Like what lowest level intensities can some insects see or other organisms? And then what portion of the electromagnetic spectrum can they see, right? Because we have a pretty limited one. We've got, 3 receptors. So we're kind of not seeing that much, right? But like a mantis shrimp, like I think they've got like, is it 8 or 12? I forget, different receptors. So they're seeing, a huge portion of the spectrum that we're not. And if there's something interacting with them, trying to hide from them, or trying to appeal to them, right, they might be modulating light out there that we just wouldn't be able to see. So that was Anthony's question. Katie asked, I wonder if temperature is important to be photoperiod. Salinity holds pretty steady around 30 parts per million, September to October. Yeah, I mean, I think temperature is a really important key here, right? And that's why we see it September, October. I think it's giving all the biological needs for this organism to bloom, right? Because Janai has great data on this, and you have it too, where we see these successional blooms happening in the lake, right? And we get this big fall one. And it's not always the same every year. It shifts as far as density of blooms, who's there, who's not there, stuff like that. But I'm guessing that warm water that we're still getting in September, October is really important because it allows for the quick, rapid development of this dinoflagellate bloom and the prey that it's eating or whatever it's eating. And then also, if this is an enzymatic process, the warmer the water is, the faster the rate of the enzymatic reactions can take place. And so this is going to tend for you to be able to do these like kind of more energetic things, like maybe run a luciferase system. But that's a lot of like what ifs in there. Anthony asks, I assume that bioluminescent light is influenced by the local habitat environment they're in and can change if the environment changes. Also, how does the environment influence how bioluminescent light is being viewed by others? Yeah, environment is a big factor there. So we kind of just talked about with that in Katie's question, right? Temperature probably has a really big influence on, I would imagine, intensity. The warmer you are, the faster you're going to be able to drive these luciferase reactions. You'll get more light at once. And then if you're thinking about this in like grand scale environment, yeah, that's probably a big factor is temperature stuff. If you're thinking about this at a chemistry level, chemical environment, like what other molecules are around, are going to be really important because they can actually modulate the light too, if they've also got ring structures. And so there are some papers when it comes to UV fluorescence, they'd notice this, that your chemical environment around the chemistry that's actually doing the UV fluorescence can modulate the wavelength a little bit. What kind of receptors, also this is Anthony's next follow-up, what kind of receptors do they have to capture the UV light? Also skin. Anthony, if you could unmute real quick and tell me which organism we were talking about here. I forget where in the talk this was.
When you first went to UV fluorescence and what you were explaining that they were I guess taking in the UV light and then producing whatever. So are they, how is that? And I think later you mentioned something, it seemed like maybe skin on the surface, they're able to absorb it somehow. I know in in understanding I'm a beekeeper, so understanding bees and that they have both compound eyes and use ocelli
Hmm.
that actually are used to detect UV light for flight and everything else. I was curious as to, so then how are they capturing the UV light and then, using it? Is it something on their body, a receptor on their body that detects it or?
Yeah, I mean, organism to organism, I think, the best studied ones are probably going to be bees and probably scorpions at this point, where there are some of these extracellular, extra ocular sort of sensory systems that are picking
Yeah.
stuff up. And they apparently, they're not doing from what we can tell, they're not doing a lot of, how would you put this? Like in the case of scorpion, it's not like they're like being able to make like then a mental map per se of like what that light is. It's just telling them, am I exposed or not?
Mhm.
And then we see this with a lot of like the bees, right? Like a lot of this is like, did something just come over the top of me, right? Am I exposed? Am I hidden? Things like that. But then this is like a place there's like a lot of research I think that could still be done.
Yeah. Okay.
Let's see here. Next, Gene, I remember seeing a lot of fluorescence in various rocks picked up in old mine rock piles in New Jersey. Is there a similarity in chemical basis? Sort of, in that you've got some sort of chemical structure that can absorb the UV light and then let go of light at a lower wavelength. But within a lot of these rocks, you're actually talking about some sort of crystalline structure that's doing that. Where within these biological systems, it tends to be these kind of complex organic molecules with a lot of ring structures in them that seem to be doing a lot of the UV fluorescence. And actually, just to throw it out there, since we're already on minerals, everybody should know California's state gem is a gem that this is the way you find it. It is UV fluorescent. It's one of the few gems I ever think about being UV fluorescent, which is benitoite. And so it's only found in two places in the world, California and Pakistan. And it's mainly because it is strongly kind of like smashed up serpentine. So you need to have a lot of fault action and a lot of active faults with serpentine around in order to form this mineral. And so down in like San Benito County is where you'd find it. There's like one or two mines you can go there to, or you can try and walk old waterways with a UV torch at night looking for this mineral. But usually it's a pretty nice gem. It's kind of like a purplish color, but then under UV light, it's got this really cool iridescent blue glow to it. But yeah, mineral structure, crystalline structure versus organic kind of highly ringed compounds is the difference between what's driving UV fluorescence in these different materials. Anthony asks, are bioluminescent fungi, insects, plants, fish, and other species edible by humans or other species, or are they poisonous? UV fluorescence, not really a good tool, and bioluminescence, not really a good tool for understanding edibility. Especially in the fungal world, there are no good rules of thumb for any eating fungi. You just want to be able to identify something properly down to a species. And no ifs, ands, or buts about that. No good rules of thumb. And it's mainly because, these things are doing bioluminescence and UV fluorescence for things unrelated to probably their edibility. Any commercial or research value of bioluminescence? There is some commercial research in bioluminescence. People are, of course, always interested in having something that glows. And so, way back in the day, right, people would take Pinella stipis, that the bitter oysterline, right, that thing that glows really well on the East Coast, but not so well here. They would take the mycelium of that, which also glows. People would sometimes call that foxfire. And they would put it in a jar and hold on to it. So like Ben Franklin has stories about this where he would use that as kind of like a nightlight. It was used in the first submarines in the world in the Civil War in order to light the inside of the submarines. The Greeks talk about also basically taking wood from this and taking it back in order to have a light at night. So there's definitely commercial, quote unquote, commercial uses that have been used for a long time, but there are people actually trying to take this to a new level. There is a group called Firefly, or is it Light Bio, and they have a product called Firefly Petunia, where they've moved the entire bioluminescent pathway, and I think it's like 8 genes, out of a really brightly bioluminescent mushroom from Brazil and moved it into a petunia plant. And so you have this petunia that like you can basically like turn, have it growing in your house and turn the lights off, and it's like producing a pretty substantial amount of light compared to other kind of bioluminescent like plant systems that I've seen. So it's definitely, there's definitely commercial and research stuff going on around that, especially because bioluminescence and UV fluorescence in the strict biology sense, these are great reporter systems. And so that's why you see the green fluorescent protein being used in a lot of like basic biology research, because it's a really nice reporter. I can put it next to something in a genome, know when it's being turned on, or know where it goes in the organism and things like that. So like GFP was really like majorly important for us understanding like developmental biology. So one of the model organisms in developmental biology is C. elegans, it's roundworm. And we know from when you get a fertilized egg where every single cell goes. And that was through using GFP to trace where cells go. So from when you go from one fertilized cell all the way out to the 939 cells of the mature adult, we know who divides and who goes where throughout that entire process due to being able to trace them with GFP. So these definitely made their way into the research world because they're very, very useful reporter systems. Recommendations on the handheld UV lights. Once again, the C8 styled 365 nanometer lights are the ones I would really recommend, just because they'll cause more things to fluoresce, because they'll get rid of the blue and purple spectrum from the LED. Debbie, microfasting else, the neotenic larva form females have the light. Thank you for that that nuance. So the males do not have it at that larval stage. Emily, Jeremy, any non-emitting chemicals that could be in an invisibility cloak. And this is one that I think about a lot, Katie, which is, I think sometimes when I see UV fluorescence, what it's telling me is that there's a compound there that's absorbing UV light, right? And so if I am, say, something that is preyed upon by something that sees more in the UV spectrum, what would be a great thing to do is actually to absorb the UV light and then kick it out in a spectrum that my predator doesn't see in. I haven't found any scientific papers around that, but I would be very surprised that if somebody doesn't bump into something like this down the road, because biology makes use of just about anything it can hang its hat on. And so I would assume being able to modulate light into a different spectrum could be used for this.
I read something when I was reading about the scorpions. No, I was reading about bioluminescent plankton, and it was talking about how sometimes the bioluminescence was not emanating just from single cells, but it was kind of put out into the matrix, which kind of made it a little bit of a camouflage for species that were trying to get the the individual cells, but they were swimming in a bioluminescent kind of soup, and that made it hard for them to focus on it. That was, I can't really, I will try to find where I read that. I was trying to read up on this topic, and I thought that was kind of an intriguing idea.
Yeah, that would be interesting, because then like if the predator's like, I'm trying to bite this thing in a certain color, and it's just basically biting nothings, right?
Yeah,
Like,
there's a
find the thing. Yeah, or digest thing.
Individual selection versus group selection. It could be a very interesting discussion. Yeah.
And see, Rivetal has an interesting one that kind of, sorry if I'm saying your name incorrectly, it has an interesting question kind of on the backside of this. Do you know about organisms that harbor bioluminescent bacteria, like the photoblaropheron fish, which lives in tropical waters, that has like a patch under its eye? yeah, the marine symbiont in a lot of these cases tends to be a Vibrio genus of bacteria. Vibrio, if you talk to a microbiologist, usually everyone's like, oh, Vibrio. What do they do with that? Because like most of our contact with Vibrio in literature tends to be Vibrio cholera, which gives you cholera. Vibrio vulnificus, which is like if you've ever had shellfish poisoning, that's usually what you've had. It also gives you like those crazy marine sores that occasionally kill people, that you get in brackish waters. But not all Vibrio are bad. There are a lot of Vibrio that do some really cool things. Vibrio fischeri is probably the best described Vibrio that produces light, and they do it in a really interesting way. And these are some of the ones that will live in the eye patches. These are some of the ones that live in the bobtailed squid in Hawaii. And they're interesting in that if you're like an organism and you want to produce light and you've got to make a friendship with this bacteria, you probably don't want the bacteria being there too much, right? Because they might get hungry and eat you, or you've got to figure out some way to deal with having this thing that's growing on you without it going bad for you. So a lot of these organs tend to allow the Vibrio to grow up to a certain concentration where they do quorum sensing. So the bacteria will get to a certain concentration and then they change their metabolism and then they'll do the bioluminescence. And then the fish or the squid will use that. And then once we're going into daylight hours, I don't want to keep all those bacteria there because they're still trying to multiply, right? I don't want them to take me over. They'll basically be able to pump the bacteria out of their organ. So squirt it out, but there'll still be a couple in there, right? And so the bacteria will start to regrow, but they're not glowing yet. And they'll basically, by the time you get to nighttime again or the darkness again, they'll regrow to a density where then you'll get the bioluminescence due to the quorum sensing. And this is really important because this is actually a very interesting model system now being used by Bonnie Bassler, who's a phenomenal scientist, to understand how does quorum sensing work? How do bacteria use chemical signals to talk to each other to modulate their metabolism? And this is really important because a lot of our disease states are caused by quorum sensing that produce biofilms and things like that. So cavities, right? This is a biofilm. Acne is a biofilm. Cystic fibrosis, usually what kills the patients is a biofilm of Pseudomonas getting in the lungs. And so if we can learn how these systems work and disrupt them, we'll have new tools to fight different types of infections that harm humans. Let's see here. Let's keep going on the list here. Another one, people are using bioluminescent bacteria to test for toxic substances in water, and there's a big industry of kits. Yeah, very, very good point. And this will be a really cool thing that you see a lot more of because you can, now that we know a lot of the molecular underpinnings, we can take those more or less cassette of genes and then move them into systems where we could be like, I want to detect this. And when I detect this, I will now produce GFP so I can shine UV light, see fluorescence, or just produce bioluminescence. So we'll get these kind of biological detector systems that we're building in the kind of synthetic biology world that's emerging right now. Gene splice spiders that generate iridescent silk, definitely in the world and possible. Yep. Anthony asks, I assume there is not much bioluminescent activity occurring in freshwater or the Arctic or Antarctica. Also, how is climate change impacting it? I think there actually is a decent amount of bioluminescence that takes place in some of these cold water systems, if I remember right. I don't have one off the top of my head, but I think there are occurrences of this in cold water zones. I vaguely remember like a friend from Alaska mentioning this. Climate change is probably impacting this in the same way that it's just impacting species biogeography in a lot of ways, right? Pushing things out, allowing new things to establish. So this is going to modulate some of these bioluminescent events that we see. Let's see here. I was looking in Amazon, found Woods lamp, which we use as a shelter to detect ringworm of cats. Yep, that would probably work. Any other ones here? We've got one new one. Oh, that's just Katie replying to somebody. I think that's all the questions I saw in the chat. If I missed something, please feel free to unmute at this point and ask, and I'll do my best to field your question. If not, I'll take a sip. Oh, go for it, Katie. You're on mute.
Yeah, I just wanted to point out that we have that really great record of plankton blooms and presence in Lake Merritt, owing to community scientists with Rotary Nature Center Friends, because our students collected the monthly samples, and then I sent them to the CDPH lab in Richmond, and they were analyzed, and they were the professional person, Matt Scanlon, was able to provide us with a detailed profile of the numbers and types of plankton that were there in each of the samples. Unfortunately, they won't do that anymore because we never found very many, if any at all, of the ones of concern for shellfish poisoning, which is why that's a mission of their lab. But I just wanted to give credit to both Matt and to all of our students who did the collecting and following of that.
Yeah, and I mean, it's really amazing what Katie and some of the other kind of, I would say, facilitators of community science have done at Lake Merritt, because even though we don't have like a formal large academic group that works on Lake Merritt, we've got these amazing data sets that we can use to kind of correlate things that we do see with things that are going on. And the ability of like Katie and others to network with people that can take their expertise and apply it to some of the things that we see, has been a boon for building our understanding of this, like, the jewel of Oakland. So thank you, Katie, for all of your years of work and keeping students motivated and looking at the lake.
That's kind of our focus with Rotary Nature Center Friends, is to connect, real science with the local person and with the incredible, as you've shown so many times, diversity and interesting questions that Lake Merritt brings to the fore for everybody, not just here, but in broader questions that that have come up. So thank you.
Yeah. Looks like we are out of questions. So unless anybody else has anything else, I think
we're just about at the usual quitting time, but this has been amazing, Damon. I cannot thank you enough. And you'll share some of the links and the resources that you mentioned in chat, so I can send the post chat thing out, email out to everybody.
Yep, I'll throw the slide deck in there one more time for anybody that missed it the first time I went in, but there's the slide deck there.
Oh yeah, I missed it.
And then actually I'll do this real quick too. I'll throw the UV fluorescent project from INAT also into the chat. So if anyone wants to kind of dig through that. That one's got the most data in it just because there's a lot more things that do UV fluorescence than things that do bioluminescence that we can see. So that's just kind of a cool one to play with. I know anytime before I go someplace new to go on a hike, especially a night hike, I will look through other people's data that are in the project and be like, what should I keep my eye out for? And priming using iNaturalist is something I really encourage people to do anytime before they go for a walk, because just knowing that something might be there increases your chance of seeing it at least fivefold.
Absolutely. I mean, that's such a wonderful resource. And you can always go back to it to answer more questions and test hypotheses and so forth. It is incredible.
And it helps people like me realize how fallible my own memory is, because I'll be like, oh, I saw that then. And then I look up at my natural and I was like, oh, no, that was a couple more years back. Sorry, Anthony, go ahead.
Yeah. So you mentioned iNaturalist, and obviously it puts out the information as to where things are. Have you encountered anything where that's a threat to these species, where people go and try to dig them up?
It's something that iNaturalist has been concerned about ever since its founding. And so they auto obscure anything that has IUCN Red List, or if a curator has said, hey, this is something that has a threat to it. So anytime something gets identified like this, we're going to obscure the coordinates.
Okay. Yeah.
Yep.
Yeah, because I would think that would be could be a problem.
Yep. And then as an individual too, you can decide whether or not to have your coordinates like open to everybody, obscured, or private. And so, a lot of like my friends that like are into like mushroom hunting, like they want to be able to like contribute the data, but not say give up their mushroom spot. And so they'll use like obscured data all the time, which is nice as a scientist looking at this, because I can understand phenology I don't care like where you're picking your, porcini at.
Yeah.
Yeah.
Well, everyone, this has been a wonderful night and wonderful questions. And I want to thank Damon and all of you for being here. And with that, let's all give a round of applause to Damon and to all of us for our interest and our passion for science and for understanding our natural world. And
And if you guys are interested in this, I will have an article out in Bay Nature in the winter magazine around UV fluorescence. So keep an eye out for that. And Bay Nature is done. What I love about Bay Nature is they run you through the hoops on fact checking, which is great. And so that will be a nice article that kind of condenses some of the stuff we went over today, and then specifically looking at just the things you'll see in the SF Bay. So look out for that in the winter magazine of Bay Nature.
Fantastic. Okay, everybody. With that, it's time to say goodnight and have a wonderful weekend, everybody. Thank you again for being here, and we'll see you next in December for another great talk. Okay, take care, everyone.
See you, everybody.
Thanks,
Thank
Damon.
you.